So, Bismillahirrahmanirrahim. In this video we will cover the last part of the lippincott biochemistry chapter number 8, and we will talk about pyruvate reduction to lactate. So now this pyruvate, it can get reduced to the lactate by lactate dehydrogenase enzyme, but they are saying that why does this lactate get produced from pyruvate? This gets produced from pyruvate because of anaerobic glycolysis in the eukaryotic cells. Now, the final fate of this lactate, um, or this pyruvate has the final fate of lactate in poorly vascularized tissue. It means that in the tissues where oxygen is less, anaerobic glycolysis will take place. What will happen in such a situation, like in lens, in the cornea of the eye, in the kidney, medulla, in the RBCs without mitochondria, this pyruvate will get converted into lactate. Now they are saying, for example, you have exercising muscle. In an exercising muscle, what happens? That you are your cells, they are less on oxygen, they have less amount of oxygen. So glyceraldehyde 3 phosphate dehydrogenase, that is producing this NADH. Similarly, the electron transport chain that is producing this NADH. What do I mean by this? If you remember the last cycle, in this reaction NADH was being produced. So ultimately it means if you have more amount of NADH as compared to NAD plus, what will happen? This pyruvate will get converted into lactate. This ratio of more NADH prefers the conversion of pyruvate to lactate in the muscle cells. With intense exercise, this lactate accumulates because the NADH is increased by both of these glycolysis and electron transport chain. Whenever this lactate increases, you should know that lactate is kind of an acid, lactic acid, so it decreases the pH of intracellular substances. And when the pH is decreased, what happens? You get cramps. That is why when you run faster for a longer period of time, you get muscle cramps all over your body. And what happens further? This lactate that is continuously being produced in your muscle cell, now it goes out into the bloodstream. From the bloodstream, it goes into the liver, and through the means of glyconeogenesis, we can get glucose produced from it. So we'll talk about gluconeogenesis in some other video. I will be making the videos on that topic as well. If you want me to make videos, please let me know in the comments below. Now, lactate utilization. This lactate, first of all, as I told you, when it goes to the liver via the bloodstream, that is happening over here, this can get converted into pyruvate, and this pyruvate gets converted into glucose, that is gluconeogenesis. Second thing is, this pyruvate can get converted into acetyl coenzyme A and go into the citric acid cycle, or the tricarboxylic acid cycle, or the Krebs cycle, as we know. Now, when it talks to the when the lactate goes to the heart cell, now it can be converted only to carbon dioxide and water through this citric acid cycle or the Krebs cycle. So utilization in liver is either conversion to glucose, either in going into Krebs cycle. The heart, it will only send it to the Krebs cycle to convert it into carbon dioxide and water. What is lactic acidosis? It is the same thing that whenever lactate increases in your body, or lactate is increased in the plasma, this is this situation, this lactate increasing in the plasma, it happens mostly. This was the normal situation, like in running muscles, but when there's a shutdown of the circulatory system, when you have myocardial infarction, when you have pulmonary embolism, uncontrolled hemorrhage, excessive blood loss, you have shock, what happens? All the cells in your body, they are not supplied with enough blood. They are not supplied with enough blood, so they're not supplied with enough oxygen. That causes them to convert from the aerobic type of glycolysis to anaerobic one, and hence they start producing lactate in larger amounts. Low oxygen supply to the tissue causes anaerobic glycolysis shifting, and hence ATP synthesis also decreases. Also, the type of lactate or lactic acid, it starts to increase. Now, in clinics, when you are looking at a patient of all these situations and you analyze their lactic acid blood levels, you can take out, or you can know the amount of oxygen that they are having, that is the less amount of oxygen in their blood. In such a situation, you can adjust the amount of oxygen that you need to give them per minute. So this is a very important clinical based scenario. Now, if you've watched my previous video in this playlist of glycolysis, you know the energy yield. I have clearly explained how aerobic glycolysis produces this much ATPs and how anaerobic one produces only 2 ATPs and no NADH. Why does it this happen? Because the glucose, it gets converted into this pyruvate, and pyruvate gets converted into lactate, but no ATP is produced. So this type of situation occurs in anaerobic one, but aerobic one yields this 2 ATP net and 2 NADH, but it yields only 2 ATP. Now we can talk about hormonal regulation. Very much easy this thing is that why do you want glycolysis to take place when you have more glucose? So when do you have more glucose? When you have taken a carbohydrate rich meal. There's in Whenever there's increased amount of glucose, increased insulin will be produced, and this insulin causes increased enzyme production or increased synthesis of glycolytic enzymes. So what do I mean by this? Let me take you to this diagram over here. So insulin, whenever it is more in amount, it will cause the synthesis of this glucokinase, this phosphofructokinase, and this pyruvate kinase. All these irreversible steps, they are catalyzed by insulin. But when you are starving, you have low amount of glucose in your body, you don't want the glucose to break down. That is why this glucagon, it is going to inhibit the whole glycolysis process, but it will cause the shifting from glycolysis to gluconeogenesis, that is now the production of glucose. We will discuss this step in another videos inshallah, but now stick to the topic that more glucose, we want more glucose to break. When you have more glucose, you want the glucose to be broken down, and more glucose causes more insulin production and hence more synthesis of these enzymes. But glucagon means you are starving, you don't want the existing glucose to break down, and hence you shut down the glycolysis process. So this was the whole mechanism. Now we will talk about the fates of pyruvate. Until now we were discussing that this pyruvate gets converted into lactate, but apart from this it also has other fates. It can get converted into acetyl coenzyme A by pyruvate decarboxylase. The pyruvate decarboxylase or pyruvate dehydrogenase complex, it is very important because it will remove carbon dioxide and also it will remove the hydrogen. So it's a dehydrogenase plus a decarboxylase type of reaction. You remove carbon dioxide, you remove hydrogen, hydrogen taken up by NADH, and hence you get your acetyl coenzyme A. This step is also irreversible, and this whole thing, the acetyl coenzyme A, is very important for tricarboxylic acid cycle. And it takes place in the cells which have high oxidative capacity, like the cardiac muscle cells. Understood? But now this pyruvate dehydrogenase complex, it is itself inhibited by acetyl coenzyme A. So pyruvate to acetyl coenzyme A by pyruvate dehydrogenase complex, which not only dehydrogenates it but also decarboxylates it. Understood? Easy peasy. Now, what if you add carbon dioxide into this pyruvate? You get oxaloacetate. So pyruvate carboxylase adds carbon dioxide, gets oxaloacetate. It is also an irreversible reaction, but now this is catalyzed by acetyl coenzyme A. So acetyl coenzyme A inhibits decarboxylation, but it supports carboxylation. Or you can remember it in such a way. It has acetyl coenzyme A has C in its name, so it will cause carboxylation to take place, or it will support carboxylation. Simple type of clue to remember this. Now this pyruvate can also get reducted to ethanol, but you know this does not occur in humans. This occurs in yeast and other microorganisms, and this is a thiamine pyrophosphate dependent pathway. So now what happens? It is a two-step process. Firstly, pyruvate gets converted into acetaldehyde and then it gets converted into ethanol. Now this acetaldehyde is a very close relative of this acetyl coenzyme A. And how do we produce it's a quiz. How do we produce acetyl coenzyme A from pyruvate? By decarboxylation. So similar decarboxylation. It also produces acetaldehyde. This is a pyruvate decarboxylase enzyme. Similarly, acetaldehyde is produced, then acetaldehyde gets converted into ethanol by being reduced. This is a reduction reaction. Why? Because hydrogen is being added to it. So this was it all. Pyruvate to acetaldehyde by pyruvate decarboxylase, and acetaldehyde to ethanol by pyruvate, or by its kind of reduction using NADH and producing NAD+. So the fates were either decarboxylation producing acetyl coenzyme A, either carboxylation producing oxaloacetate, either reduction, this kind of reduction that occurs not in humans but other microorganisms, producing decarboxylating and producing acetaldehyde, reducing and producing ethanol. So this was all of the whole of the chapter number 8 from your lippincott. I hope you understood each and everything. You can if you have any questions related to this chapter, please ask me in the comments below, and we will meet in some other videos. Till then, we have completed the chapter number 8, and I am very much happy that we did it together. Yes, we did it together. Uh, and let's meet in another video discussing something else, inshallah. Please like, share, and subscribe to my channel to watch more good stuff like this, inshallah.